Welcome back, friends. We'll be talking about protein trafficking and different modes and aspects of protein trafficking in very basic way, not in the detailed way, because I've already talked about each of these different stages of protein trafficking detailed manner in my different videos. You can watch them in my YouTube channel. Now, so let's begin with this protein trafficking concept. Now, first is the overview of sorting of nuclear-ended proteins. Now, we know that proteins can be of different types. Proteins can be delivered outside the cell. They can be sent inside the nucleus. Sometimes they are transferred into some organelle like mitochondria or chloroplast or peroxisome. Sometimes protein must be transported from one region of the cell to another region of the cell for sorting. For example, once it is made inside the endoplasmic reticulum, it should be taken out for the sorting of the proteins for the delivery into the sorting house, which is called the Golgi bodies of the cell. So it creates the total Golgi network. That's also very important for sorting out different varieties of protein, from the protein to be secreted and the proteins to be delivered to the organelles, all of these things. But in this video it is shown the nuclear delivery proteins. That means those proteins who are ended up inside the nucleus and uh, inside different organelles, right, and some other secretory pathways. So it's all the different varieties of protein. If you look at here in first case, that from mRNA it is translated into protein product. Now once it is translated into protein product, at the end of it, it is having a signal that's called the signal sequence. Now the signal sequence can be of different type. For example, it is a ER signal sequence. Once it receives the ER signal sequence, that protein molecule will enter inside the endoplasmic reticulum, because that's what the signal sequence is telling the protein, right, to enter inside the ER on the endoplasmic reticulum. So it is now inside the ER. Then it will be taken into the Golgi bodies, because ER and Golgi, they are cross-talking with each other inside the cell all the time. So those proteins will be bulged out as a vehicle, as a vesicle-like structure. It will fuse to make the trans Golgi, then the medial Golgi, then cis Golgi, and finally they will bring those proteins, to modify those proteins inside the Golgi bodies. For example, the modification can be uh, glycosylations, and many different types of modifications can be taken place. Then after that, the proteins are, once proteins are modified and prepared for delivery, they will be taken outside the cell, and they can also be delivered into other sac-like structures like lysosome, or they can be simply docked into the plasma membrane and the protein can be released. Now this is a basic way for the secretory pathway of proteins, because some proteins are secretory, they must be secreted outside the cell. This is a way for the for producing and delivering secretory proteins. Now other ways, for example, if we want our protein to be ended up inside the organelle like mitochondria or plastida like chloroplast, so in that case what we need some different signals like mitochondrial signal or mitochondrial association signal, mitochondrial signal sequence, or chloroplast signal sequence at the end of it. So once it is getting mitochondrial or chloroplast signal sequence at its ends, as you can see in this case in this yellow colored thing, right, so it's a peroxisome signal sequence specific, right, organelle specific target sequence should be there at the end. Now that target sequence will be received by the different receptors that are present onto the surface or sometimes inside the organelle. Once they receive that signal, they take this protein inside their organelle and place it, for example, the proteins of mitochondria or chloroplast or peroxisome. Now, in other cases, as we know that the organelle specific uptaking of the sequence is important in case of nucleus, because they should have the nuclear signal sequence, or uh, NS S, right? So nuclear signal sequence is very much important, because using those nuclear signal sequence, those proteins should be delivered inside the nucleus through the nuclear pore, right? So in this way, many different varieties of proteins are made and they are transferred from one region to another region, and that's how this whole process actually works. Now in the future slides we'll be talking about each of those important stages actually. uh, okay, so let's begin. So here we can see the specific uptake targeting sequences. You don't need to mug up and memorize all of these things, as I don't encourage you to to look for that. But actually, if you look at here, that the endoplasmic reticulum is having the signal in most of it, in most of these signals are of N-terminal end. So the signals are actually present in the N-terminal end most of the cases, right? So if you look at here, that the endoplasmic reticulum signal is also present in N-terminal end. The mitochondrial signal sequence is also present at the N-terminal end. For the chloroplast, it's also present in N-terminal end. For peroxisome, it's present in the C-terminal ends, and so on. So depends on the type of organelle they are targeting, right? Most of them, they are present in the N-terminal ends, because once the protein start to synthesize, the N-terminal end is the first type of region which is start synthesizing, and so on. The first six sequence, like 6 to 10, 12, 15 amino acid long stretch of the sequence, become that kind of signal region, which will be recognized by the different receptors, protein molecules present in organs, and all these things. So if we look at here, except for peroxisome, rest of them are having the signals in the N-terminal ends. And if you look at here, that they are having the signal removal portion, yes or no, both of these things are there. And if you look at here, the nature of signal can be varied. For example, the nature of signal can be hydrophobic amino acids, it can be hydrophilic amino acid, the stretch of 2 or 3 amino acids repeatedly, like arginine or lysine residues, or hydrophobic amino acids, and many different variety of amino acids are repeatedly present sometimes to give this characteristic signal sequence a strong site. Now, if you look at here, the translocation of the a protein into the mitochondria, because I've told you that we'll be talking about each of those different sections independently. So if you look at here, the translocation into a mitochondria, and how does it actually work? That this nest. So normally, the idea is that we are having an unfolded protein, or slightly partially folded protein in the cytoplasm, and we need to deliver it inside the mitochondria, because if we fold this protein outside the mitochondria in the cytoplasm, then it won't probably end up inside the mitochondria, because remember, in all these cases, this proteins are, once it's folded, it will receive its characteristic structure, and once it receives its characteristic structure, it will be very difficult for the protein to pass through the pores that are present in this organelle, or in the membrane of the mitochondria and all these things. Okay, so for that reason, what we need to do, we need to deliver these proteins in an unfolded conformation inside the organelle first, then it is getting folded inside that organelle, or inside that destination region more specifically. So if you look at here, this is the nascent precursor molecule, so we need to guide it through a channel inside the organelle, and then we need to fold it. So there are three major tasks to be done. First task is to guide it in such a way so that it is not getting destroyed by the cytoplasm, because if there are any hydrophobic residues present in the protein, that can be hampered by the cytoplasmic presence, because cytoplasm is mostly made up with water. So for that reason, we need to take this protein, and you can see that at the N-terminal end there is a signal that's called the mitochondrial signal sequence. Now, the first important thing is that there are certain proteins that are guiding a protein from one region to another region, and those proteins are called chaperonine, the helper protein. They help the proteins to be not to be misfolded actually. So for example, you can see the cytosolic HSC70, it's actually called HSP whatever. So this is a kind of protein. Once it is bound with our nascent polypeptide here, our polypeptide is now good to go, but addition of this cytosolic HSC70 requires the hydrolysis of ATP, because it requires energy. Once this process is done in all these cases, you can see that once it's done, it can directly put this nascent polypeptide onto the receptor present in the outer membrane of mitochondria, that's called TOM, right? So actually there are two types of complexes that guide these proteins to be entered inside the mitochondria. One is the TOM, which present in the outer membrane, another one is TEAM, which present in the inner membrane. So you can see here the TOM. Now the TOM here is present in the outer membrane, TEAM actually presents in the inner membrane. So sorry, so let me take a color for you, because it will be required in this case. Okay, so if you look at here, that this is the TOM, so it can directly bind with TOM, and then it can transfer it into the TEAM, which is present in inner membrane, and then it can enter this protein inside the mitochondria. But if you look at here, the other stage is via another protein that's called MSF, or mitochondrial signal receptor response element. That element is another protein molecule which will recognize the signal or mitochondrial signal sequence that's present in the N-terminal region of this nascent polypeptide, and it will recognize it, it will bind with it, and once it's bound with it, it also requires energy. You can see the ATP hydrolysis is providing the energy in that case also, and then this MSF will transfer this nascent peptide to further complex called TOM70 and TOM37. But we've seen in the previous cases, if it directly interact with HSC70, it will transfer it directly into the TOM40, right? So actual way is first TOM70, TOM37. In that case, it need to be transferred into TOM40, then through the channel into TIM17, and then it will be entered inside, right? But if it's using this HSC70, cytosolic HSC70, then the process is one step less. That's the binding and the action of MSF. Now once it is entered inside the mitochondria, through that place they again require some other HSC protein, HSC70 proteins, and these are called matrix HSC70 proteins because they are present in the mitochondrial matrix. So this mitochondrial matrix HSC70 proteins again will use some ATP to be attached with this peptide that is there, and then once bound with it, it will take this inside, and then it will be added with some other HSC70 proteins which will help and guide this nascent peptide to be folded inside the mitochondria, and then we get our active proteins inside. As you can see here, HSC70 is required, some more proteins are required, and energy is required. So throughout this process it requires a lot of energy, like ATP hydrolysis in this stage, and many different stages this hydrolysis of ATP is required, right? So this is a way of how a protein is transferred from a cytosol inside the mitochondria. Now the translocation of proteins into chloroplasts. Now if you look at here, the translocation of protein into chloroplast, the basic idea is the same, that it should have a nascent polypeptide which is having a signal sequence destined to deliver the protein inside the chloroplast. That's called the stromatal import sequence. As you can see here, the stromatal import sequence, or SIS stomatal import sequence is there. Actually I have a kind of technical fault in this case. Anyways, so it should have this stromatal import sequence at the N-terminal end. Once it is having this, it will bound with the transporter that is present, the channel that is present onto the membrane of chloroplast, and that channel is simply termed as TOC. Now different variety of the TOC protein, like TOC75, TOC34, TOC86, and all of them. Actually TOC86 is acting as a helper protein there. Majorly it is composed of TOC75, and TOC34 is a GTP associated proteins, or GTP associated proteins, that is guiding the protein to be inserted inside. So once it is having this subunit, for example say Rubisco, so let's say this is a protein called Rubisco S unit. Actually Rubisco is having two different subunits, S subunit and an L. S stands for small subunit, L stands for large subunit. The large subunit mitochondria actually chloroplast produces inside itself, but the small subunit must be transferred from the cytosol. So this is the small subunit of Rubisco enzyme, and it is now having the stromatal import sequence. It will be guided through this TOC75 channel using ATP hydrolysis because energy is required. Once the nascent polypeptide is inside the stroma, which is a matrix of chloroplast, as you all know. Now once it is inside the stroma, it will involve the HSC70 proteins again, because these are chaperonine, because these are very important proteins, guide the other proteins to be folded. So they will guide this nascent polypeptide to be folded, and for this folding they require again ATP hydrolysis. So ATP will hydrolyze and it will fold this nascent peptide into a particular structure, and during this process they will also cleave this terminal amino acid sequence, which is the signal sequence. They will cleave the signal sequence out of it, and during this process this small subunit will be attached with the Rubisco large subunit, which is also being guided by another chaperonine called HSC60 to be folded properly. Then these two folded agent, one is folded using HSC70, which is the small unit coming from outside of the chloroplast, coming from cytosol, and the large subunit is folded or guided the folding using the HSC60 or chaperonine that is present inside the stroma, and then both of the subunits are attached with each other. They are arranged together via the, again using the energy of ATP hydrolysis, and we get the assembled product, right? So that's how the whole process can work out like this. Okay, now let's talk about how the proteins are translocated from cytosol into another organelle like peroxisome. Because the processes are, though the idea is kind of same, but processes are slightly different, guys. Because the translocation into the peroxisome, if you look at here, the targeting of protein is initiated actually the post-translationally, actually, because once the protein is completely translated, because it's all true for the translocation of proteins into mitochondria, the translocation of protein into chloroplast, and this the same thing is for peroxisomes also. So once the proteins are made, but the simple difference is there because the peroxisome is also require a sequence which is telling the protein to be inserted into the peroxisome, not the any other kind of organelle, and that sequence is called the peroxisome signal sequence or peroxisomal protein signal sequence, PTSIR. So PTS actually, the peroxisomal transmission sequence actually, and it is also required another protein called PTSIR, which is a receptor or protein receptor of that signal sequence, which will bind with that PTS sequence, and then it will help that protein to be inserted into the peroxisome. But actually the uniqueness for the peroxisome is that the peroxisome usually contain uh, this signal sequence in its C-terminal end, not in not in the N-terminal end, because we have seen in case of mitochondria or chloroplast, this whole sequences are present in the N-terminal end, but in case of peroxisome, they present in the C-terminal, as you can see here. Okay, this is the signal protein, right? And once this process once it is sequenced with the signal sequence at the C-terminal end, uh, heme or iron groups are required, because iron group will arrange four iron group are there, should this four iron group should be arranged with the unfolded sequence of nascent polypeptide, and they will form a kind of catalase tetramer. Once the catalase tetramer is formed there, once the catalase tetramer is formed, then you can see that using that PTSIR protein, that PTSIR protein will be attached with any one of these signal sequences actually in this case, right? So once it is bound with this, then PTSIR protein will guide this whole iron and protein complex or heme protein complex inside the peroxisome through the channel created by a protein called PUX or PEX14 actually. So this PEX14 is a receptor, it's a protein receptor that is very important for binding with PTSIR element, and PTSIR, once it's bound with it, then it will deliver this whole complex of iron protein cluster inside the cell, right? So once it is inside the cell, then again once after inserting into the lumen of peroxisome, that PTSIR protein dissociates from the rest of the sequence, and this PTSIR is now free, as you can see here, it is now free again for the next round of its work, right? So that's how the whole process works in case of the translocation into the peroxisomes. Okay, now we need what we need to know. We need to know the overview of the whole secretory pathway and how it's actually works. So very basic, guys, is for a layman how this process actually is done. Now this whole process is actually called the cisternal progression, and in this case actually the idea is very simple, that the proteins are made and they are injected inside the endoplasmic reticulum, right? And that endoplasmic reticulum is termed as rough endoplasmic reticulum. So here you come with the mRNA, and ribosomes are sitting onto it, and proteins are polypeptides are made, they're inserted inside the ER or rough ER, and once inside the rough ER, those polypeptide chains are getting folded, and once they're folded there, once in the folded condition, they will be taken out from the ER as vesicles, right? So as you can see here, they are taken out as vesicle. Many different varieties of proteins are there, like one blue, one red, green and black, many different types. Some secretory, some should be delivered into organelles, and so on. So once they are there, they will migrate into the cis Golgi reticulum and cis Golgi network. This is the first type of Golgi network that they are encountering from the synthesis of ER. Now through this cis Golgi network it will migrate to cis Golgi, medial Golgi, trans Golgi, and finally the trans Golgi reticulum. And this reticulum part of the Golgi bodies, they are kind of fragile because new vesicles are docking, they are making fusion, and they are elongating it. So, and from the trans Golgi reticulum new vesicles are coming out, they are going there and attached with the membrane, and they are elongating the membranes, spanning they are expanding the membrane, as well as they're delivering the proteins outside. So here you can see, suppose in this case this black protein should be delivered into the lysosome. So you can see after the sorting event that's done here in this Golgi apparatus, this black molecule is delivered into the lysosome. And in the similar way, if the red proteins and blue proteins are those type of proteins which should be delivered outside the cell, and they are sorted inside the Golgi body through the trans Golgi reticulum. Finally they produces like they produce the secretory vesicles. Inside the vesicles there are those proteins, and once those proteins are inside, those proteins will be taken and those vesicles will be fused into the plasma membrane, and all of these proteins will be released outside of the cell. And once they're released outside of the cell, they can be inserted into the membrane, or they can be directly outside of the cell, right? So in this case you can see the secretion process can be of two different type, like one is the regulated secretion, another one is the constitutive secretion. Now the constitutive secretion is simply the secretion in that case, in which case we simply deliberately constantly delivering protein molecules inside the vesicle from the Golgi outside the cell. And the regulated secretion, we should receive some signal from outside via any means, and then after the response of the signal, our proteins will be delivered outside, right? That's the whole process. Now analysis of yeast mutants to define the different major stages in the secretory pathway. So that's a very important thing, because for the protein analysis we usually remind and rely on the yeast cells, because yeast cells are typical small cells, and they are eukaryotic cells, so very small eukaryotic cells, easy to work with. So that's why we choose yeast always in that cases. So you can see here that we can analyze yeast cells in many different ways. Like, as you can see, that delivery of the yeast cells in the normal secretion, what happens, we are having proteins that are inside the ER, then they are in the Golgi, then they are in the vesicles, then they are released. Now in case of class A type of proteins, as you can see here, they are produced and they are present and accumulated in the cytosol, right? And the class B type of proteins, the accumulation in the rough endoplasmic reticulum, because the proteins are synthesized and they stay in the rough endoplasmic reticulum for longer period of time, right? So why we can see these different stages, because we hamper different important proteins inside the cell. Suppose if we hamper the transport of protein into the ER, the proteins will be made and they are distributed into the cytosol, right? This thing will occur. Now what will occur if we block the budding of vesicle from the rough endoplasmic reticulum? The proteins, once they are made, they will be pushed inside the ER lumen, but the function of budding out of the from the ER lumen is blocked. As a result, a huge amount of proteins will build up, and it will kind of fill the ER lumen inside, right? Now if we block the fusion of transport vesicles with Golgi, now if that thing is blocked, then the accumulation of this protein will be there in ER to Golgi transport vesicles, because neither the protein will be seen in ER nor the protein will be seen in Golgi, because the docking of the vesicle with the Golgi is blocked, and already the proteins are released from ER. So all of those proteins will be present inside the cell, inside the vesicles, right? So that thing we can find. So this will give you the idea of the whole process of protein trafficking and how the protein trafficking is working in different stages of protein trafficking. If we block different functions, it will give us different results. For example, if we block the transport of Golgi to secretory vesicles, suppose the proteins are delivered into the Golgi apparatus, but we block the delivery and the sorting activity of Golgi and the production of vesicles from the Golgi. In that case, the proteins will accumulate in the Golgi, right? So this thing will result. And finally, if we block the transport of the secretory vesicles into the cell surface, right? So actually the final stage, the secretory vesicles should be coming out from Golgi, and they will be attached with the vesicle for the protein to be released. If you block those vesicles, then those vesicles will start to accumulate inside the cell, but there will be no delivery of proteins outside the cell. So that thing will occur there. So there are many 5 different classes of proteins, that protein mutants actually, or situations, defective function mutants actually observed using yeast cells, because secretion is very much seen in the yeast. It's very common and kind of very basic and similar like eukaryotic, most of the eukaryotic cells. Now if you look at here the translocation into the ER, what we can see here. So it's a kind of detailed stage, because translocation inside the ER is the first common thing that should occur, right? So for that reason actually the translocation can occur usually, most commonly, is a co-translation translocation. That means ribosome is sitting onto the ER, onto the membrane of rough endoplasmic reticulum, or RER, as we all know, RER. Very sorry because it's kind of giving me trouble, so it's very sorry whether presentations are going wrong in this case, which is the co-translational translocation of RER. So in that case what we know that so let me erase the background. Okay, so what we can see here, that co-translation translocation is there, which is ribosome is synthesizing the polypeptide chain, and as the polypeptide chain is synthesizing, it is inserting itself into the ER through the ER uptake complex, which is made up with a protein called Sec61 complex, right? So this is called co-translational translocation, because as these proteins are translocated during the translation process, that's why it's called co-translational translocation, because both these things are going on simultaneously at the same time, right? But again they require certain proteins like SRP, or signal receptor protein, or signal response sequence protein. So those proteins will help and guide this nascent polypeptide chain, which is just formed and synthesized from ribosomes and just coming out of the ribosomes. And there are protein receptors called SRP receptors that are present onto the surface of ER membrane, ER membrane, and that SRP will interact with the SRP receptor, and then they will transfer this whole polypeptide chain along with the signal sequence, which is here in the pink color, inside the ER lumen. In case of other way of transferring this is a post-translational translocation. In the post-translational translocation, once the protein is completely synthesized inside the cytosol, which is outside the ER obviously, then this protein will be taken by a helper protein onto the Sec complex, or the transfer complex called the Sec61, Sec62, which is this transfer complex here, right? So for that reason, so that protein, that helper protein, is called BIP, right, or BIP protein. So this protein will be taken, it will attach with the nascent polypeptide, it will bring the nascent polypeptide to another protein called Sec63. Now once they are bound with Sec63, then they will transfer this nascent peptide, actually the signal sequence, onto the Sec61, Sec62 complex. Once that's done, then BIP will cleave the nascent chain from its signal sequence. As you can see here, it will cleave the nascent chain from its signal sequence, and this stage of transferring this nascent chain from the Sec61, 62 complex to this BIP protein required ATP, right? So once ATP is required, they will cleave it out, and then the protein will be inserted into the ER. And we know we don't need to bother about the folding of the protein inside the ER, because ER is designated to do that. It is having all the necessary machineries to do that inside. So it's kind of very easy, guys, and that's how it's done. Now, if we look at the topologies of some integral membrane proteins, and they actually synthesized via ER. So many different varieties are actually there. You can see a variety where it is called the anchored kind of protein. It's called the glycophorin, for example, or LDL receptor, or you can see the example of influenza HA protein. All of these proteins, they're kind of embedded via helical domain into the membrane of cell. And the C terminal is in outside, and N terminal is inside the cytoplasm. Sorry, C terminal is inside the cytoplasm, and inside is outside the cytoplasm, right? Now if you look at here another one where the same thing is there, but but N terminal is towards the cytoplasm, and C is towards outside the cytoplasm, and that's called that's the type of transferrin receptors or Golgi galactosyl transferase enzyme. Now, if you look at here, cytochrome B5 or cytochrome P40, the structures are kind of similar but slightly different, because you can see here that again N-terminal is towards the cytosol in that case, but the whole part of the N-terminal, which is very simple for both this glycophorin and this transferrin receptors, as you can see. But in case of the cytochrome receptors, the part that is connecting outside is very small, right? And there are proteins which are spanning the membrane several different times, and that's called the 7 transmembrane type of proteins. Most of the signal receptors are like those. G protein signal receptors are like those. Or example is GLUT or GLAT, whatever, glucose transporter protein, that's also present onto the cytoplasm, embedded onto the cytoplasm, just like stitching thread onto the onto a okay. So the final one, this is the bacteriorhodopsin. That's again several transmembrane domain, actually 7 transmembrane domain, or 7 TM type of protein, 7 transmembrane type of proteins there, right? And also you can find it in the beta adrenergic receptors, right? And in one terminal we are having so C terminal is facing cytosol, N terminal is facing outside the cytosol. But in case of the in case of GLAT receptor, you can see it's not actually 7 transmembrane, it's multiple transmembrane domain containing protein, and both the N and C terminal facing the cytosolic region. So that's the two major difference between the GLAT and the bacteriorhodopsin, because bacteriorhodopsin are 7 transmembrane, spanning the membrane 7 times, but GLAT transferring it many different times. And in GLAT you can see both them in N terminal and C terminal at the cytosol. But in case of bacteriorhodopsin or beta adrenergic receptor, it is only one. So C terminal in the cytosol, towards cytosol, N terminal is towards, or it's outside the cytosol. Okay guys, welcome back. And in this video we'll be knowing that what are the different types of modification and sorting that is going on inside Golgi apparatus, because you all know that inside the Golgi apparatus modifications have taken place and the proteins are sorted and their chemical modifications are done. So what are the types of modification that take place inside the Golgi apparatus? That's what we need to know here. Now normally what we know here that the different types of construction or breaking down of proteins modification can be taking place, like glycosylation, sumoylation, methylation, acetylation, and many different types of changes can be there. Now two such changes are noted here. One is the production of a functional protein, which is albumin, we all know, from its precursor called proalbumin. So here you can see this is the proalbumin, this is the albumin. So actually proalbumin, when synthesized inside the ER and then transported from ER into the Golgi apparatus, it contains 2 arginine residues at the N terminal end, right? So at the N terminal, so 2 arginine consecutively. Now this 2 arginine is a kind of sequence that is preventing the rest of the molecules to be active, right? Now once it is taken inside the Golgi apparatus, these 2 arginine residues are cleaved away. Once they are cleaved away via the endoprotease enzyme, that's called the furin endoprotease enzyme. Once these 2 arginines are cleaved using furin endoprotease enzyme, rest of the parts are free. Now this rest of this part, which is designated here with the only red color, is termed as albumin. Now it is an active part of the protein. It can function as albumin protein, whatever it is. Okay. Now similarly, the second example is of production of hormone, very important hormone, important for regulating the blood glucose level, which is insulin. Now once insulin is made, it is a kind of long large chain. As you can see here, it's a large chain having N terminal, C terminal domain. It is having different residues in between, that's arginine, 2 consecutive arginine, lysine arginine residues are there, and it is also having sulfur units coming out of different sections. Now the sulfur units that we've seen here that are coming out from different sections, these units are due to the presence of cysteine, right? So these sulfur units are present and there is sulfur bridge or sulfur bond between them. right? So sulfur disulfide bridges are present between these sections, right? Now what we know that normally when this insulin present in this state, it's called the pro-insulin. It's a previous function form of insulin. It is not active in nature. Now what we see here, normally we require some endopeptidase. It's called the PC2 endopeptidase and PC3 endopeptidase. This PC2 or PC3 endopeptidase, not a peptidase, actually endoprotease that can cleave the sequences from the terminal region, from this one from this arginine sequence, one from this sequence, and ultimately it gives us first the PC2 endoprotease works and PC3 endoprotease, both of them work together, and as a result of that it will cleave away this section including lysine and arginine residue, and it will give us a semi-functional kind of insulin. Not actually semi-functional, but still the insulin is not non-functional. It's not totally functional now, till now. After that, the second round of cleavage will take place, and this will be assisted by carboxypeptidase. Like so, previously endopeptidase, now carboxypeptidase will work. Now carboxypeptidase will cleave the two arginine residues from here, and as it cleaves two arginine residues from here, it will cleave these residues out. As a result, rest of the parts are arranged with each other. They are attached via sulfide linkages, right? Like two different units, unit A, unit B of the insulin, they are held together, and rest of the materials, rest of the amino acids like arginine, lysine, whatever present in between, now fully cleaved away. First the cleaving of via PC2 and 3 endoprotease, and then the carboxyprotease. So two round of cleavage is taking place to convert proinsulin into active form, which is insulin. Okay, now let's talk about another very important function of this vesicle transport inside our body. It's a real life example, guys. Till now we have talked about the schematic presentation and how this process may occur and what are the different mechanisms for this process to occur, but this time it is a real life example, and the example of the LDL receptor binds and internalizes the cholesterol containing particle, because you know, once inside our body the fat molecules, whatever you eat after the digestion, that's present there. Now those fat molecules can consisting of cholesterol esters, mostly made up with cholesterol, a huge amount of cholesterol. It is designated here with this yellow colored section, and there are phospholipid layers. So phospholipid is kind of masking the whole cholesterol ester in such a way. You can see here phospholipid monolayer, and in between those phospholipid monolayer there is unesterified cholesterol present, unesterified cholesterol that is embedded inside, embedded between. And also another protein which is a helper protein, which is bound with this cholesterol ester and this whole mass of cholesterol and phospholipid monolayer, that's called Apob protein. Now Apob protein is the protein which helps this complete structure of cholesterol ester and unesterified cholesterol, whole this lipid content to be delivered from our intestinal lumen through our liver, and it actually goes into a liver and the liver cells actually, right? So how this process is actually done, so if you look at here this whole system along with this red Apob protein, as you can see here in this triangular shape, so this molecule will come and actually inside onto the surface of our cells. Actually we have different LDL receptor, or low LDL means low density lipoprotein, because there are many variety of lipoprotein, low density, high density, and many varieties. So low density lipoprotein receptors or LDL receptors are present onto the surface of the membrane, which will bind with this Apob protein, interact with Apob protein. Once they are interacting with Apob protein, it will bring several different small molecules like clathrine now. So clathrine molecule will come, they will arrange through or surrounding this LDL receptor inside the membrane which is facing towards the cytosol. Now it will completely cover this up, and it will kind of engulf, it will help this whole construction of LDL, this whole lipid molecules along with the Apob protein to be internalized inside. Once it is inside of this coated vesicle, now this coated vesicle, from this coated vesicle, clathrin proteins will be recircularized onto the membrane for the future round of uptake, but rest of the structure is uptaken via vesicle formation. That's called early endosome. Now once the early endosome is there, it will be fused with late endosomes or sometimes fused with lysosomes. And once they're fused with lysosome or endosome, actually the early endosome will now fuse with late endosome, and the pH of this environment changes. Now you can see the pH of 5 or less than 5. It's slightly acidic in the range. And finally, once it is bound with, so actually once it is bound with the late endosome, which is having a slightly lower pH than the early endosome, the rest of the structures that we can see, that is the LDL receptors, are cleaved away from the LDL and whole cholesterol structure, whole cholesterol mass, and those LDL receptors are recycled again onto the cell membrane, right? And rest of the part is now inside the late endoplasmic late endosome vesicle, late endosomal vesicle. Now those late endosomal vesicle will be fused with the transport vesicle. Now the transport vesicle, actually here it consists of lysosomes. So lysosomes now will be bound with will be fusing with this late endosomes, and after the fusion, these lysosomal enzymes will degrade rest of these proteins. Will actually break down all of these molecules apart, like they will break down this cholesterol esters apart, and unesterified cholesterols apart, phospholipid monolayers apart, and Apob protein, everything. They just cleave those things apart, and we get all of these things independent, like cholesterol, fatty acids, amino acids, and rest of the parts. Okay, and then it will be transferred into the required region where we need to transfer them. Now this is a third, and this is another very important stage. It's actually two unique setup where we've seen that the process of transport, the process of protein trafficking occurs two unique ways for two very special purposes. One purpose is to deliver immunoglobulins from the intestinal fluid, from the blood and intestinal fluid, into the intestinal lumen, actually. So immunoglobulins are very important, because in different infections, actually cells need to look for whether there is an infection or not, and if there is an infection, then they need to supply a lot of immunoglobulins, as well as they need to make sure that whatever is inside our intestinal lumen, intestinal lining of the cells, they are correct, they are right, because a lot of things are going on in intestine there. So actually this process called transcytosis, and we will be seeing this machinery, as well as we are going to see that this transport system or membrane transport system is actually helping not to deliver proteins, but also to deliver some uh, element, like this element is iron in this case, right? And using a protein receptor, protein helper called transferrin, right? So it's called ferrotransferrin, a protein molecule called transferrin, and there's iron attached to that transferrin, converting it into the ferrotransferrin molecule. Now the ferrotransferrin molecule, that is protein along with iron residues, will come and attach with the ferrotransferrin receptor, or transferrin receptor rather, present onto the surface of the cell, onto the surface of, which is the membrane, onto the cell. Now once they are bound with this ferrotransferrin, bound with the transferrin receptor, they will be internalized via endocytosis pathway, and clathrin will help them to coat this whole vesicle. That's the clathrin coated receptor mediated endocytosis pathway. So once this process is done, then after this process, after the internalization is done, the clathrin molecules will be recycled, and rest of the section, that's the endosome, will be there. Early endosome will be converted into late endosome, and once it is converted into late endosome, the pH suddenly drops, and all of those transferrin receptors are now free, and they will be recycled. And then what we know, finally once it is ready, it will fuse with the late endosome, and as the pH drops much, like pH less than 5 or 5, in that condition, those ferrotransferrin molecules, ferrotransferrin proteins will become apotransferrin, and all of these irons are just coming out of those proteins, because they are the construction of protein and iron present in between the protein structure, protein cleft. So each of those proteins are containing two irons. So here you can see irons are coming out, and those ferrotransferrin are now converted into apotransferrin molecules, and then the irons are taken out of this late endosome into the cytosol, and that's how a cell can get iron. It can take iron through a protein molecule, but not via channel transport. Actually, it's via a receptor mediated endocytosis pathway. Slightly unique, but it is present. Similarly, if you look at here, the blood lining and the intestinal lumen. So in intestinal lumen, actually there are several things. There are food materials, water, everything is coming in and out. In this case, sometimes infections may occur, many different kinds of infection. If there is any kind of infection or certain things that we can see, in that case they need to check it. Now if there is any infection, there should be the presence of antibodies that are present. So for checking that antibodies present there or not, what they are having specialized kind of cells actually. Now those cells are actually having all this interaction between the immune cells along with the intestinal lumen, right? So they are called kind of M cells, right? So here we can see that normally here in there is an endosomal transport machinery with which they can kind of producing uh, receptors, FC receptors, a kind of antibody receptors onto the surface of the intestinal lumen cells towards the intestinal lumen. Now there are also immunoglobulins like immunoglobulin G. Now immunoglobulin G, along with the help of its FC receptor, can attach with the receptor with the FC portion, can attach with the FC receptor that is just being added onto the surface of intestinal epithelial cells, right? So as you can see here, that's the thing which is required, because if anything is going on, if antibodies that are present inside the intestinal lumen, using this FC region and FC receptor onto the surface of this luminal membranal cells, then they can uptake those immunoglobulins inside, right? That's how this whole trafficking can be going on. And not only that, but also sometimes bacteria, not only immunoglobulins here, but also some bacteria. If there's any bacterial infection, bacteria can be present in intestinal lumen. In that case, bacteria act as a kind of antigen in that case, and it will be attached with some kind of receptor that is present onto the surface of these M cells that are present there in the intestinal lining, and they will take up those cells and they'll package them, and they will transfer it directly to the immune system cells that are present onto the other side of this epithelial cells into the blood lining. Now those cells are macrophages and many other cells. Those macrophage and other type of cells will kill the bacteria containing cell, and that's how they can actually sort out and figure out, is there any kind of infection going on into the intestinal lumen or not. So these are the two different ways, and this machinery is called transcytosis because it's kind of endocytosis pathway, but it is transferring from one side to other side of a particular cell lining. That's why it's called transcytosis of some ligands across the cell. So these are two unique properties that we can see inside our body, but both of them are important: transfer of iron as well as the checking whether there is any infection in the intestinal lumen or not. So that's it guys, and I hope that's helpful. Thank you.